Hello and welcome to this lesson about the digestive system. And today we're going to learn about the structure of the digestive system. Also we'll learn about the organs of the digestive system. And also we'll learn about the adaptations of the small intestine. But before we move on to that, I think it would be helpful if we defined digestion. Now, digestion is the breakdown of large food molecules into smaller ones that can be absorbed and assimilated. And what that means is that when we eat food, it's usually too big. The molecules are usually macromolecules, they're too big to be absorbed into the bloodstream, so we've got to break them down first, then they're absorbed, and then whatever the molecules that we eat, they're turned into human molecules. For example, we eat animal protein, that's turned into human protein. So that's digestion. It's the breakdown of large food molecules into small ones that can be absorbed and assimilated. And the next point that we're going to go on to is the structure of the digestive system. And I'll attempt to draw a diagram. And just a word before I start, if you need to pause the video at any point to recap a diagram or a definition, then please feel free to do so. So in a digestive system, what we've got is we've got the mouth, which I've just represented here, down into the esophagus, into the stomach, and then into the small intestine, and the small intestine is usually quite tricky to draw. So I've simplified it slightly. And then into the large intestine, which does a lap of the small intestine, if you like. That's the large intestine going around the outside there. And then the rectum. And then finally the anus at the bottom of the diagram. Quite appropriate that the anus is at the bottom. I'm just going to add a few other parts there. I've just drawn the gallbladder and the pancreas in there, and then the dotted line that I've drawn there represents the liver. So we'll label it up. This is the mouth. This is the esophagus. I've used the American spelling there. This is the stomach. This is a simplified version of the small intestine. This is the large intestine. This is the rectum. And this is the anus at the bottom. And that is the path of food through the body. The food actually passes through those organs: mouth, esophagus, stomach, small intestine. The first 15 centimetres of the small intestine are quite important, and we call those the duodenum. We'll come back to that part of the small intestine a bit later. And then you've got the rest of the small intestine, and then the large intestine, and then rectum, anus. But there are other parts of the digestive system which the food doesn't directly pass through, but are nevertheless very important. You've got the gallbladder here. And that, what the gallbladder does, is it stores bile which has been produced by the liver. And then you've got this organ, which is the multifunction organ. It's called the pancreas. So the pancreas is very important. We'll come back to that again. So that is an overview of the digestive system and the organs within it. And what we're going to learn is we're going to learn about the role of those organs in the digestion of starch, which is a polysaccharide, proteins, and lipid. Alright? And we'll start with starch. Starch digestion begins in the mouth. And the mouth has salivary glands which add salivary amylase to the food before it goes down the esophagus and then into the stomach. And that salivary amylase will begin the digestion of starch. And what it does is it digests starch into the disaccharide maltose. Now that digestion is really quite brief, because when the food gets into the stomach, as we know the stomach has a very low pH, it produces hydrochloric acid. That salivary amylase is actually denatured. So it is quite a brief digestion, and starch digestion doesn't restart until we get into the small intestine. And what happens in the small intestine, I'll just draw it here. This is the pancreas, and the gallbladder goes off there. We're not really interested in that at this stage. And this is the duodenum, the first 15 centimetres of the small intestine. The pancreas is here. Take a leaf-shaped organ. The pancreas produces pancreatic amylase. So the amylase enzyme is produced by the pancreas. It's mixed with the food. Pancreas also produces sodium hydrogen carbonate, which will neutralize the stomach acid, which allows that pancreatic amylase to work. That pancreatic amylase then continues starch digestion for any starch that wasn't digested in the mouth. And that completes the digestion of starch into maltose, and the maltose will be digested a bit later on further down the small intestine. That's starch dealt with. Protein digestion takes place in the stomach itself, and if this is the stomach, just for the sake of completeness, you've got two circular muscles at the top and the bottom of the stomach called sphincter muscles, the esophageal and the pyloric sphincter. So food can be retained in the stomach. And protein digestion takes place in the stomach. And if you have a look at the wall of the stomach, like so, the esophageal and the pyloric sphincter there, apologies if they were off the screen briefly. If you have a look at the wall of the stomach there, if you zoom in on the wall of the stomach, what you'll see is these pits called gastric pits. And they are, like every other part of the body, they are made up of cells. And those cells, to be fair, quite diverse. They secrete different things, and as all cells have nuclei, I'll just add some nuclei here. There we are. And these cells secrete different things into the stomach. Some are enzymes, some are not. For example, you've got this cell here. It's called a goblet cell. It secretes mucus. You've got this cell here. This cell secretes pepsinogen. And then you've got other cells which secrete hydrochloric acid. So these cells of the gastric pits secrete the things that we need to digest protein. Now just a word about pepsinogen and HCl. Quite an interesting relationship between the two. Pepsinogen is an inactive precursor of pepsin. And pepsin is a protease enzyme, but when it's secreted as pepsinogen. Now if you think about it, that's quite sensible. Because if pepsinogen is an inactive precursor, what that means is that it's not going to break down the cells themselves. In fact, it stops something called autodigestion. And autodigestion would cause damage to cells, and it could also cause stomach ulcers, so we definitely do not want any autodigestion. So we secrete, the cells make pepsinogen, which is an inactive precursor, which is then activated by the hydrochloric acid which is present in the stomach acid. And that starts off protein digestion. And how pepsin works is if you've got a protein there with tertiary structure, what pepsin does is it acts like a pair of scissors, if you like. So it'll cut. Let's see if I can get this right. That's not bad. It cuts the protein up basically by hydrolyzing peptide bonds between specific amino acids, and that will take you from this protein with tertiary structure to much shorter polypeptide chains. And that's what the action of pepsin, that's what pepsin does. And then you've got your protein into a stage where it can then move down the rest of the small intestine, and we'll be returning to protein digestion in a little while. The final bit of digestion that we're going to have a look at is lipid digestion, or fat digestion. Now, lipids aren't soluble and they form droplets. So the first thing we've got to do to that lipid is what's called emulsify it. And emulsification, you won't be surprised to hear because I go on about it all the time, is increasing surface area, and it's quite important. And what emulsification does is it takes large fat droplets and it breaks them up into much smaller droplets. And that increases the surface area and allows lipase enzymes to act. And how that happens is we're back to the duodenum again, but this time we are interested in the gallbladder. So this is back up to the first 15 centimeters of the small intestine. And the gallbladder stores bile which has been produced by the liver. And that bile comes down this duct here, and then it is added to the food which is coming down the small intestine. And the function of bile is to emulsify fat and increase its surface area. So after that fat's been emulsified, and we've made these small droplets, like I said before, then the lipase enzymes, which I've drawn like little circles with the active sites there, they can then get at that lipid and break it down into glycerol and fatty acids, which can then be absorbed not into the bloodstream but into the lymphatic system. So that's fat digestion as well. So we'll move on now to the adaptations of the small intestine itself. Now, the small intestine, the function of it is absorption. A very important function, because there's no point in eating something if you can't absorb it. We are very well adapted to get as much nutrition from the food we eat as possible. We evolved at a time when food was scarce, and so we make the best use of any food that we eat. And the small intestine is very well adapted for this. If you do a cross-section through small intestine, what you'll find is that the walls of that small intestine are folded. And the folds themselves, if you were to zoom in on them, are folded even further. And that second level of folding, they are called villi. Alright? And we're going to zoom in on one of those and have a look at the detail of it, because those structures, the villi, are very well adapted to carry out absorption and to complete the digestive process. So let's have a look at one of them in a bit more detail. This is one villus. And as we know, they're made up of cells. All of these are epithelial cells, which you may have heard of when we did the cells topic, and they all have nuclei. So I'll just put all the nuclei in each one of these epithelial cells. Every cell in the human body has a nucleus, with the possible exception of red blood cells, which are packed full of hemoglobin, so they don't have nuclei. So that's the basic structure of the villus. Now, inside each villus, you've got a network of capillaries. The blood comes in, and then the capillaries branch out, and then the blood returns. And I'm going to put the return section of the capillary bed in blue. Blood doesn't just change colour from blue to red, but it happens much more gradually than that. But it helps us to see in this capillary bed that the blood is coming in this way here, flowing past the food and then flowing out. It actually goes to the liver next. And you can see that this provides, and I'll write it down because it's quite important, a very short diffusion pathway. Because the middle of the very middle of the villus here, the bit where the blood is very close to what's called the lumen of the small intestine, and the lumen is the middle bit, that's where the food is. So you can see there's a very short diffusion pathway there. So digested nutrients, such as glucose and amino acids, they've got a very, very short diffusion pathway to get into the bloodstream there. But that's not the only thing that's present in the villus. We've also got a lymphatic vessel, which comes up here. And this is a branch of the lymphatic system. Because not all things, particularly if they're not soluble in water, can be transported by the blood. So you've got here this structure which contains lymph, and that is where fat is transported, and that structure, that's called a lacteal. So that is a villus. And I'll just label it up here. We've got epithelial cells. We've got a capillary here. And it's a pretty good diagram, and probably a good time to have a sip of coffee. And if you need to pause the video, just have a look at the diagram in a bit more detail, then feel free to do so. Now, moving on from that, we can even zoom in further and have a look at one epithelial cell. Alright? And that's what I'm going to do next. So if you have a look at one epithelial cell, on this side of the epithelial cell, we've got the lumen, which is where the food is, lumen of the small intestine, and on this side of the epithelial cell, we've got the blood, which we saw in our capillary there. So digested nutrients diffuse, some are actively transported from the lumen into the blood, and you can see these structures here. These are called microvilli. And they increase the surface area even further. So we've seen that there's a lot of different levels of zoom on our diagrams here, and I'll just go through them in quick succession. We've got the folds in the small intestine. Those folds are folded into villi. If you zoom in on one villus, it looks like this. And if you zoom in on one of those epithelial cells, you can see that it's folded even further into microvilli to increase the surface area. So you've got maximum surface area for absorption there. The other thing that everyone always forgets for another adaptation of the small intestine is it's very long. I mean, I drew it with just a few changes of direction there, but you can see that the food spends quite a long time in the small intestine before it enters the large intestine and then exits the body. And that's time in which the nutrients can be absorbed from it. So the small intestine is very well adapted for the absorption of nutrients. Now, if we zoom in even further onto these membranes themselves, we'll find out where digestion is actually completed. Because on those membranes of these cells, these epithelial cells in the small intestine, what you see is you can see membrane-bound proteins. The membrane-bound protein here. And if you remember, this is a phospholipid bilayer. Just quickly fill that in. And you can probably guess what these proteins are. These are enzymes. And this would be the active site of the enzyme, this section here. And the substrates which will be binding to the active site would be things like that we mentioned before, like for example maltose will get broken down into maltose will get broken down into glucose by the action of the enzyme maltase. And also another enzyme, you've probably heard of maltose to be honest, but another example of an enzyme which you'll find on an epithelial cell are enzymes called exopeptidases. And those enzymes, exopeptidases, they break peptide bonds between amino acids, and they finish off the digestion of protein. So then when you've got amino acids and you've got glucose and you've got fatty acids and glycerol, then they can diffuse across these cells and then be transported either by the capillaries or the lacteal, which is contained in the villi. So just to recap what we have done, our objective today was to get an overview of the digestive system, and I think we've done that. We've got a digestive system there, and the organs of it are outlined quite nicely. So you know the structure of the digestive system and the structure of the small intestine. We can name the organs, the main organs of the digestive system. The ones that the food passed through: the mouth, the esophagus, the stomach, the duodenum, small intestine, large intestine, rectum, and then anus. And the other organs which are involved, namely the gallbladder, which is situated in the liver, which stores bile, and the pancreas, which produces pancreatic amylase, pancreatic lipase, and also pancreatic protease enzymes. So the pancreas is really a multifunction organ. And then we went on and we looked at the structure of the small intestine, which we've zoomed in on here to look at these brush border enzymes. So I hope you've learned something from this video. I might make a few more of these after I'm finished, and thanks for watching.